For L10c, we'll talk about transcriptional control, the basal transcription apparatus, transcriptional activation. An important example of transcriptional control, the phosphoenol pyruvate carboxykinase gene. Then we'll talk about ribosomal genes, transcription and processing, class 3 genes, tRNA and 5S ribosomal RNA, and then we'll talk about microRNA, abbreviated miRNA. So we've already talked about basal transcription and forming the transcriptional complex at the promoter. So we had the Tata box binding protein, TF2D, TFA, TF2A, TF2F, BE, RNA polymerase 2. Here's our TF2H, which remember was a helicase and a protein kinase. And this is called the basal transcription apparatus. And in genes that are controlled by this complex, we have what's often called constitutive RNA synthesis, and we have constant levels of low to moderate levels of RNA that's made during most stages of the cell cycle. And then we have inducible transcription. And inducible transcription, you have some sort of activator complex that will activate the basal transcription apparatus. Upon this activation, you actually recruit more RNA polymerase 2s and you increase the level of transcription of genes. So in inducible transcription, we have what's called an enhancer, and this is a binding site for an activator protein. Then we'll have an activator protein. These activator proteins can be steroid receptors or other type receptors that bind to these enhancer sequences, and upon binding to the enhancer sequences, notice they make a loop. It may well be that these particular kind of genes and activators are involved in loop domains. We may not have to make extreme loops like that. This strand of DNA may be proximal to this strand of DNA to begin with. So here we have a histone acetylase involved in this, chromatin remodeling complex involved in this, and then here's our basal transcription apparatus. Here's the mediator interacting with the basal transcription apparatus. These are particular proteins that are going to relax our nucleosomal structure and allow transcription. So binding of general transcription factors, RNA polymerase, the mediator, this is the mediator, it binds to the activator protein complex, chromatin remodeling, histone acetylases. Regulation of phosphoenol pyruvate carboxykinase gene expression by glucagon and glucocorticoids. Remember, when blood glucose is low, the liver synthesizes glucose by gluconeogenesis, and this is controlled by glucagon and glucocorticoids. Glucagon, remember, activates protein kinase A through cyclic AMP, and we have a protein that's called CRIB, and CRIB means cyclic AMP response element binding protein, and it becomes phosphorylated. Here's the cyclic AMP response element. So this is the part of the promoter that's activated by cyclic AMP. Glucocorticoids are steroids, and they bind to the glucocorticoid response element. And here's the glucocorticoid receptor complexes. This is called hepatocyte nuclear factor 3. Since we're in the liver, this is what makes it tissue specific. It's this hepatocyte nuclear factor 3. Here's our basal transcription apparatus. Upon binding of the glucocorticoid receptor to its response element, we swing the DNA strands together. There will be an adapter protein in here somewhere. It's not shown. And then, upon making this structure, we start the phosphoenol pyruvate carboxykinase transcription. So here is RNA polymerase 2. Here's the nascent pre-mRNA that's being synthesized. So both cyclic AMP and glucocorticoids activate the transcription of the phosphoenol pyruvate carboxykinase gene. And now we know that when glucose gets high, insulin inhibits the phosphoenol pyruvate carboxykinase gene and the protein. We can have high glucocorticoids. Here's the receptor binding to the GRE. We can have cyclic AMP. Here's CRIB, although it's not phosphorylated. Assume it's phosphorylated. And this is called the insulin response sequence binding protein. This is the insulin response sequence. And so insulin, through its tyrosine protein kinase activity, activates this protein. The protein binds to its response element, and it blocks the binding of hepatocyte nuclear factor 3. And by blocking this binding, it inhibits the formation of this activation loop, and we've inhibited the synthesis of phosphoenol pyruvate carboxykinase mRNA and the protein. And by dominant, that means we can have all three factors that are high. We can have glucocorticoids high, we can have cyclic AMP high, and we can have insulin high, and insulin will inhibit this complex. So that's what we mean by dominant. So this process right here won't happen until insulin levels decrease and glucose becomes low, and then we can form this activated complex. Very interesting gene control. Modified bases that are found in tRNAs and are ribosomal RNAs. Down here, when you look at the PowerPoint, you can read the legend that's in this particular slide, and it says there's over 100 of these modified bases that have been identified and characterized. So we have 4-thiouridine, so rather than an O, that's an S. Inosine, remember that was the first product of de novo purine biosynthesis. 1-methylguanosine, this is guanosine, and there is the methyl on nitrogen 1. Here's N6 isopentenyl adenosine, it's called I for isopentenyl, I6A, and this is the I6A. That looks like an isoprene unit. So N6 isopentenyl adenosine. Ribothymidine. Remember, we have thymidine in DNA, but in RNA, we also have thymidine in very low amounts. And here is thymidine, and rather than being hooked to deoxyribose, is hooked to ribose. Pseudouridine, this is uridine, no problem, but rather than the ribose being bound to the nitrogen, it's bound to this carbon. So that's called pseudouridine. And dihydrouridine, this is uridine, and there's this double bond right here is reduced in dihydrouridine. So first, let's talk about ribosomal RNA transcription, modification, and cleavage. And then our next transparency will also talk more about this and integrate some things. So remember, ribosomal RNAs are made by RNA polymerase 1. So we're transcribing a ribosomal gene, and we're getting a 45S precursor of ribosomal RNA, and the S of 45S, that has to do with how big the particles are. So the bigger this number, 45 is bigger than 30, 60 is bigger than 45, and it has to do with how fast they move when you centrifuge them. So the bigger they are, the faster they move, the bigger this number is. So S stands for something called Svedbergs. So we have 13,000 nucleotides in this primary transcript, have a 5' end, a 3' end. The sequences in blue are going to become ribosomal RNAs. The sequences in grays are not going to become ribosomal RNAs, and they're going to be cut out or off. In this case, they're cut off. In this case, they're cut out. These are not intervening sequences because they're not interfering with the sequences of a gene. So this entire blue part here is the gene. So these are inter-gene sequences. So there's no gene in here. There's no gene sequence in here. So they're not intervening sequences, as in introns. They're spacer sequences. They space this one from this one, from this one. So then we have chemical modifications. Some of those bases we saw on the preceding page are going to be incorporated in this mRNA. So notice we make the ribosomal RNA without the modifications, and then we modify the bases that are in the ribosomal RNA to get our modifications. And we're going to talk about what are called SNO proteins, which are small nucleolar RNAs that direct where we put these different chemical modifications. We degrade the regions of nucleotide sequence. We have cleavage, so this part gets degraded, this part, and that part are degraded, and that leaves us with an 18S ribosomal RNA. That's this part right here that now is modified. This small part is 5.8S ribosomal RNA, and then we have the 28S ribosomal RNA, the 5.8S and the 28S, plus the 5S, which is made not in the nucleolus, but in some other part in the nucleus. These three combine with a large number of proteins, and they give us a large ribosomal subunit. The 18S plus the ribosomal proteins for the small ribosomal subunit combine and give us a small ribosomal subunit, and this entire process happens in the nucleolus. So this is an overview of ribosome synthesis. So here's our loop of nucleolar organizer DNA. So this is found in the nucleolus. And remember, these ribosomal RNA genes come from our agrocentric chromosomes. You may want to look up what agrocentric is. They're the ones with short arms on the top. They look like little buds. Look sort of like insect eyes sticking out. And then we get the transcription by RNA polymerase 1, and we get the 45S ribosomal RNA precursor. Then we have our SNO RNAs, and they're going to bind to this precursor, and they direct the modifications on the ribosomal RNA, modification and processing of ribosomal RNA. And then also from the cytoplasm, we get the proteins involved in processing ribosomal RNA. And what might those be? Those are the enzymes that direct the modification. So we have RNAs plus enzymes that are being transported into the nucleus and then into the nucleolus, and they're directing our modifications. We have ribosomal proteins that are made in the cytoplasm, and they go through the nuclear pores, and then we start assembling our large ribonuclear protein particle. It's huge. It's a huge particle. Over here, as we start assembling this, all of these proteins and the SNO RNAs dissociate, and they can be used again on another precursor. Here comes our 5S ribosomal RNA, and it's coming from the non-nuclear parts of the nucleus. It combines with this large ribonuclear particle, and now we have an immature large subunit. We'll have an immature small subunit, and upon maturation, they leave the nucleus via the nuclear pores. So we actually make intact small ribosomal subunits, intact large ribosomal subunits, and this whole thing happens in the nucleolus. So the nucleolus is a ribosomal factory, and then once we get into the cytoplasm, we can have our messenger RNA. We'll assemble the 40S subunit and the 60S subunit to make the mature dimer ribosomes, which will start translating our messenger RNA. Ribosomal structure, prokaryotes versus eukaryotes. You could probably guess the prokaryotic ribosomes will be smaller. So here we have a molecular weight of 25 million. Here we have a molecular weight of 4.2 million. We have a 50S subunit, a 30S subunit. Over here, we have a 60S subunit and a 40S subunit. The large subunit here contains a 5S ribosomal RNA, a 23S ribosomal RNA, plus 34 proteins give us the large subunit. Over here, we have the 60S, which has a 5S ribosomal, a 28S ribosomal, and a 5.8 ribosomal, plus 49 proteins. So rather than 34 proteins, we have 49 proteins. Here on the 40S, we have an 18S ribosomal subunit, plus 33 proteins, give us the intact small ribosomal subunit. So the eukaryotic ribosomes are more complex and larger than the prokaryotic ribosomes. tRNA transcription and processing. So our tRNAs are going to be produced by RNA polymerase 3. So here's our DNA, which has tRNA genes on it. Often these genes are found in tandems, and we might have the tRNA genes for many, many different tRNAs. And by many, many different, I mean different sequences, and they're going to accept different amino acids. So notice we get a transcription, and then this actually contains an intron. Why is this an intron? Because here's a coding sequence separated with an intervening sequence by another coding sequence. And notice how these are forming loops, and these particular loops have to do with this part of the molecule, base pairing with this parts of the molecule. So this is intramolecular base pairing rather than two separate strands that are base pairing. So the strand folds over and base pairs with itself. So this is base pairing, this is base pairing, this is base pairing. We're going to cut out the intron and cut off the 5' end and the 3' end. So now this is shorter. We're missing this part right here. And we're going to modify bases. An addition of CCA is going to be the part that accepts the amino acid. The part that we've joined right here, this is going to be what's called the anticodon. This is the part of the tRNA that will recognize the messenger RNA triplets. Here we have dihydrouridine, the 2 Ds, pseudouridine, ribothiamidine, so T is ribothiamidine. This is pseudouridine, this is dihydrouridine. So here's some modified bases that are in the tRNA molecule. And these are also probably directed for these modifications by SNO-type RNAs. So here's our process tRNA. This shows the structure in a little more detail. So we have a GG, pseudouridine is right here. Here's our CCA, this is the amino acid attachment site. This is called the variable loop. Different tRNAs have variable loops of different sizes. This is pseudouridine that goes in here. Ribothiamidine goes here. Here's T-Psi-C loop. So this contains ribothiamidine, pseudouridine, cytosine. So we have this loop right here. And here's our anticodon loop that's going to recognize our mRNA. Again, this is basically an expansion of what this picture here showed. So here's our primary transcript. Notice you can see now how it's base pairing with itself. GC, UA, CG, UG. So it stops base pairing when the turn gets too short and there's no base pairing between these particular bases. A and C don't pair, C and U don't pair, U and U don't pair. And remember, we can only do RNA-RNA base pairing for a short number of nucleotides. Remember, RNA DNA is about 10 nucleotides. So this shows us our primary transcript. And now here's all the modifications that we've done. So this is actually showing more modifications than we have in these particular figures. So this has dihydrouridine, dihydrouridine, dihydro. There's 5 of them. So this is the dihydrouridine loop, D loop. Here's 7-methylguanosine, 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 our CCA amino acid acceptor. Here's methyladenosine, pseudouridine, ribothiamidine. Here's another pseudouridine, another pseudouridine. So we have many, many different modified bases in tRNA structures. And again, what we've done, we've removed this part, we've removed this part, we've added the CCA, we've added our modified bases, and now we clip out this part from here to there. That gives us our loop. MicroRNA, miRNA, and control of complementary mRNA degradation. So we've talked about making mRNA, but typically your mRNA doesn't last very long. It's destroyed by a number of mechanisms. And this is one of the mechanisms that seeks out mRNA and accelerates its degradation. And I have 2 pictures, and they're basically equivalent. RNA polymerase II makes these miRNAs, and notice when we make them, these are called pre-microRNAs, and they're long, and here's a large structure, and notice we have these loops, a lot like we had the loops in transfer RNAs. And then we hydrolyze these particular loops. We basically hydrolyze between the hairpins, and we make structures that look like this. The RNase that does this particular processing to make these hairpin structures is called DROSHA, and DROSHA plus another protein recognize particular sequences in here and generate these hairpins. We have a protein that's called exportin 5, and exportin 5 binds to the RNA, and it allows it to transverse the nuclear pore. So this is one of those nuclear export factors that we saw when we talked about messenger RNA. And now once we get into the cytosol, we have another RNase activity that's called dicer, and dicer takes these particular RNA hairpins, and here's dicer binding to it, and it removes the loop structure. So now we have 2 complementary RNAs. Remember, they were one molecule that base paired with itself, and now they're two separate RNAs. These two separate RNAs can bind to a protein that's called RISC, and RISC is RNA-induced silencing complex, and it binds to RISC, and when it does that, there's a couple of things that can happen. It looks for mRNA sequences that are complementary to the miRNA. If they're very closely complementary, like a perfect match or close to a perfect match, this results in mRNA degradation. So this particular complex will direct the degradation of the mRNA. If it's almost a match, it will slow down protein synthesis. So binding with mRNA rather than degradation will slow down the reading by the ribosome. And this picture shows the same thing. Here's our RNA polymerase II. Here's the transcription. There's our pre-mRNAs, and you can see the loops, microprocessing by dosha, and another protein that's DGCR8 separates these hairpins. We have export over here. We talked about export in 5. We have the export, and now Dicer removes the loop. We unwind the two pieces in this model. One of the pieces binds to RISC. So here's the RISC loading. And then again, if we have near perfect complementarity, so here's our RNA with the poly A tail. This is the 5' end, so this must be the 3' end, the 5' end. If it's near perfect complementarity, we destroy the mRNA. If it's partial complementarity, we repress translation. So these are microRNAs, and there's literally thousands and thousands of these different sequences that are leaving the nucleus and controlling the stability and the translation rate of protein synthesis. So controlling the stability of the mRNA and controlling the rate at which the ribosomes translate the mRNA. So that completes lecture 10C, and our next class will be protein synthesis.